We have got your slides. Would you turn on your camera?
Are you hearing me?
Yeah, sure.
Like to thank Professors Bahrami, Matin, and Bithori for their kind invitation and harmonizing this valuable symposium. And I will try to talk briefly about the as the last speaker.
We have lost your voice.
Do you have my voice?
That sounds better. Yes, go ahead, please.
Yes, yes, okay. I'll try to talk briefly about the translational regenerative medicine, focusing on scientific bridge between basic knowledge and clinic. And we'll try to talk about the stem cell translational medicine pathways and bench to bedside journey, and also talk about the requirements of preclinical studies, and also GMP and GCP. And at the final, introduce some major translational barriers. As you can see here, and I named it vertical regenerative medicine life cycle, from the ancient studies and finding some evidences in the nature and the human fetus about the regeneration, to developing the first iPS and the first embryonic stem cell to recent years, that the human can produce different types of cells and tissue-specific cells from the iPS and other stem cell, multipotent or pluripotent stem cells. And beside this vertical life cycle, we have another life cycle, horizontal life cycle for development. When you have an idea about the stem cell therapy and regenerative medicine, you should translate it step by step using some restricted guidelines and standards and legislation authority mandatory guidelines to clinic, and putting an effective fingerprint to improve the global health, from the basic researches as in vitro, in vivo studies, to phase 1, 2, 3, and 4 clinical trials, and then you should translate and commercialize your product and move to the market to try to achieve a good outcome in your patients and put a good impact on and improve the global health in worldwide. But in moving from the basic researches in the stem cell and regenerative medicine to clinic, the scientists and clinicians should try to avoid some lost in translation. To avoid from the lost in translation, we need some legislations, regulations, and also authorized body supervisions. Such this slide you can see here, some example from the FDA and Europe. Advanced therapeutic medicinal product development, you can see here, when you start basic researches, you need just good laboratory practice guidelines and quality control in your laboratory. After entrance to preclinical and clinical studies, you need more and more, and you should change your atmosphere, and you should commit to some restricted guidelines such as GMP, GCP, to perform your clinical trials, and then some knowledge about the commercialization to move and enter to the market. As you can see here, as an example of our studies, at first we need to get some approvals, for example, from our ethical committees from the, for example, Ministry of Health or your universities, to start your studies in vitro or in vivo, and also especially in the clinical studies. Then you need to control the quality of your product using different methods, flow cytometry, karyotyping, or differentiation methods, and also imaging methods, to achieve the best safe and the best quality product. To start your preclinical and especially your clinical trials. In preclinical study, we have some options and items that should be considered. For example, in vivo studies are necessary for what? For demonstrate our proof of concept, for show the safety and efficacy of our stem cell product in humanized models, and also predict some technical problems that may the scientists face when perform their clinical trials. But there are some challenges. For example, what is the best and the most appropriate model, human model for your disease? And also there are some significant immune reactions and rejections, and you can use immune compromised or immune suppressed animals. And also you should design your experiment like to clinical trial design, and also define clearly your outcome measures. And before starting clinical trial, some items should be clarified. For example, the activity of your products, effective dose, regimen, and also route of administration. And also the scientists, when perform animal studies, should commit to some guidelines and also standards. For example, 3R rules: reduction of the number of the animals that you use in our researches, refine the techniques, and also replace other procedures to avoid animal study as possible. After modeling and validation of your animal model, you can implement your stem cell based product, your cell-based product in the animal, and use some imaging or other techniques to achieve the best data. These data in animal study is very important, such as infrastructure to start the organization of your clinical trial. And you can see here some imaging techniques for tracking and tracing your implanted cells in animal bodies. But after a successful animal study, you will try to start your clinical trial. As our products, most of stem cell based products are more than minimally manipulated products. We need GMP standards and facilities to start our phase 1 and then 2, 3, and 4 phases of clinical trial based on FDA, Iran FDA, and also EU GMP. For example, your product should be processed and produced in a classified standard GCMP laboratory, GCMP facility, or clean room facility. And the scientists all need to commit GMP compliant protocols in all of their procedures for producing a clinical product, not product for research use only. And you can see here some documentation that talk about clinical production of regenerative medicine based products. And as I said previously, to move from the basic science to the clinical application, we need some standards, for example, GMP, good manufacturing practice, and we need to produce our products based on pharmaceutical and GMP guidelines in classified facility under supervision of authorized bodies. After that, for transplanting or implanting our products in human, we need another standard. Its name is Good Clinical Practice, GCP. But what is the GCP? GCP is a standard for design, develop, monitoring, analyzing, recording, reporting of all of the results and data from the extracted from our clinical trials, and can guarantee credibility and accuracy of our data, and can guarantee our trial subjects and our patients' protection during performing the clinical trial and is very important part of our translational chain from the basic to the clinic. In this paper, we reported the first draft of Iranian National Guideline for Cellular Therapy Manufacturing that was developed by my authors and I in collaboration with the Iran FDA and also Iranian Stem Cell Council. After that, in 2015, the first regulation for cell tissue and gene therapy released by the Iran FDA. And in conclusion, regenerative medicine can revolutionize disease treatment. What we need to clarify and show is our product safety, efficacy prior to start the clinic. And also scientists and clinicians need to know more about the translational pathways, commercialization, and other techniques and knowledge related to the translation or the products and application of them in the clinic. And before starting the clinical studies or clinical application, some items should be addressed in our studies. For example, safety, therapeutic effects, biodistribution, immunogenicity, tumorigenicity, and other items based on our product's specificity. And we have some translation barriers in this road that I explained you. The first one is the lack of knowledge between academic members about the regulatory and also commercialization knowledge and processes. The second one is the short time perspective that is designed by academia, industry, and regulatory bodies about the regenerative medicine development and application. And the last one is the lack of the budget. There are 6 criteria to select one project for starting and developing in field of regenerative medicine like to other fields. The first one is the burden of that disease that you aim to start your project to solve some problems of the patients in field of that disease, that incurable disease. The second one is very important, is the final impact that you could put on the global health. And another ones are feasibility of and affordability of that project for development, and also acceptability of that socially, ethically, and legally, and its safety, and also some indirect benefits that select our and prioritize our project to select and start for development based on our region and country status. But here at final, I want to give you some take-home message. The first one is that investors today have difficulty understanding how regenerative medicine can be marketed. And it needs to talk more and more about commercialization of regenerative medicine with the investors and also pharmaceutical companies about the future of the regenerative medicine and future of its market. The second message is this: regenerative medicine will impact on health, but not replace the medical procedures and devices, but the scientists should gather all of the disciplines and methods to solve some more problems of the patients and human. And the last one, translational regenerative medicine is not solo, is symphony. Symphony that should be harmonized by a general population, scientists, clinicians, and regulatory bodies, and more stakeholders to achieve the best outcome and impact on the global health. And at final, I want to thanks again of the executive members of this symposium, and also apologize because I couldn't come to Mashhad and attend the symposium in the Mashhad because I am in translation from basic to clinic, because now I'm studying internal medicine since 2 years ago in Tehran, and because of the limitation of time, I'm so sorry I couldn't come to Mashhad and see our colleagues at that nice city. Thank you very much. Thank you so much for your time.
It was so great. We received some information. In the meantime, it goes to raise some questions in our mind. Thanks anyway. Hope to see you again in other coming congresses. See you then. Bye-bye.
Thank you so much. Thank you.